Hi, everybody. I just want to, again, thank you for attending the Planetary Sample Science Seminar Series that we run every Wednesday for, again, the rest of April. And this seminar series is being sponsored and hosted by Survey Central, the CASA Moon node of Survey Central, the Institute of Meteoritics, and the UNM Grand Challenge. And probably even more appropriately is today's food in room 340 of Northrop Hall is being provided by the Center for Stable Isotopes. It's my pleasure to introduce James Dottin from Brown University, who will be giving a talk on using sulfur isotopes to constrain lunar evolution.
All
James, it's
right.
all yours.
And I'm assuming you can hear me okay?
Yes.
Great. All right. Well, I'll start off by saying I'm jealous that I'm not getting the food in Northrop Hall, sponsored by the Institute for Meteoritics, but I'm glad you all are enjoying some good food. So yes, today I'm going to be giving a talk that describes some of my work where I've been using sulfur isotopes to place constraints on how the moon has evolved. So today I'll go through a couple of different projects that I've been working on and try to um, describe the next steps. So as many of us are aware, there are about four main stages of lunar evolution. It starts off with the giant moon forming impact, and then the moon goes into differentiation into a core, mantle, and crust. There's also the mantle overturn event. And then there was a significant amount of volcanism, both as effusive basaltic volcanism, but also as fire fountains. And lastly, there is the most recent stage where you have lots of solar wind, sputtering, and implantation, and micrometeorite bombardment that causes soils and grain volatiles to be moved around on the lunar surface. Now, what's important about each and every one of these stages is that they play a significant role in establishing the contents and and the cycling of volatiles. And it's important that we understand the volatile contents and how they're cycled because volatile elements on planets, they drive planetary evolution, and they also they control climates and are, at least for Earth, are super important for the preservation of life. And ultimately, as we place constraints on volatile contents and how they're cycled, we can ultimately determine various processes associated with the evolution of the moon. Now, among the six most important life-preserving volatile elements on Earth, I'm most interested in sulfur. And this is because sulfur has four stable isotopes. It also exists in multiple oxidation states in the rock record, from, say, sulfur -2 to sulfur +6. And when we combine information from both the isotopic compositions and the, I guess, petrographic or petrologic context of the rocks, when we combine those two, we find that sulfur makes an excellent tracer of planetary evolution. For example, we can use both isotopic compositions and an understanding of the oxidation state of the sulfur to determine the interplay between a planetary surface and a planet's atmosphere. Now, as I mentioned before, sulfur has four stable isotopes, 32, 33, 34, and 36. And in my work, I use the ratios of these isotopes in order to evaluate mass-dependent fractionations, which is graphically shown here as a mass-dependent fractionation line. And I also use the ratios of the isotopes in order to evaluate mass-independent fractionations. And that's graphically shown here as a deviation from mass dependence. And in this talk, you'll see two different notations. The first is the Dottin notation, which is used for describing mass-dependent fractionations, and also this capital delta 33s notation for mass-independent fractionations. Now, when I use the term mass-dependent fractionations or effects, I'm referring to fractionations that occur when the chemical or physical properties of that fractionation depend on the mass of the molecules involved. So these are properties such as the vibrations, rotations, and translations of molecules. And so some clear examples of this process, say, on the moon would be the process of, say, sulfur degassing, where you have a preferential loss of either 34s or 32s that results in a fractionation that's strictly dependent on the mass of the molecules involved. And ultimately, as we assess mass-dependent fractionations, we find that various rocks that we analyze have fingerprints of how systems were processed and how the elements within it were processed. Now, when I use the term mass-independent effects, I'm referring to fractionations that occur when the chemical or physical properties of that fractionation depend on anything but the mass of the molecules involved. So these are properties such as interaction with the electromagnetic spectrum or spin-orbit interaction. And one of the most widely observed examples of this process in the sulfur system is the process of UV photolysis. And it's been readily observed in Earth's early atmosphere, such as in the Archean. And so in my work, I use the measurements of both mass-dependent and mass-independent fractionations to constrain processes associated with understanding how sulfur has been processed on a planet's surface, within a planet's atmosphere, and also within a planet's interior. Now, in order to make sulfur isotope measurements, there are multiple methods, from both in situ to bulk methods. I have developed an expertise in performing sulfur isotope measurements using gas source mass spectrometry and fluorination. And so quickly, I'll just walk through how I have made the measurements specifically for the lunar samples that I'll show today. So first we start with a bulk soil or glass that's digested in a bath of chromium chloride, HCl, and hydrofluoric acid. And as that bath is heated, the sulfur that's in the rock is released as H2S. It's then bubbled through a water trap in order to capture any acid vapors from that reaction. And then lastly, it's bubbled through a silver nitrate trap, where that H2S then precipitates out as silver sulfide. We then go through a process of cleaning the silver sulfide and drying it, and then fluorinating it with fluorine to convert the silver sulfide to SF6. And then ultimately, we then end up purifying that SF6 because there are other contaminants that are generated in that fluorination process. And we purify that using a variety of cryogenic techniques and a gas chromatograph. And finally, we end up analyzing our purified SF6 on a Thermo Finnegan MAT 253 gas source isotope ratio mass spectrometer. So this leads me to the first part of my talk, where I'm going to be discussing a project that we recently published in Geochemical Perspective Letters, where we looked at the sulfur isotope composition of lunar soils in order to understand how volatile elements are cycled on the lunar surface. So lunar soils, they represent a relatively volatile rich reservoir on the moon. They consist of the sub-centimeter fraction of the lunar regolith, that's been gardened through micrometeorite impacts and solar wind interactions. And they are composed of mineral and rock fragments, breccia fragments, glass, and agglutinates. Now, what's interesting from the transcripts from the Apollo 17 mission is that there's potentially some evidence that the soils on the lunar surface are coated with a condensed layer of vapor that has a possible reactive sulfur component. So for example, if we look at the transcript, we see, oh man, does that smell, doesn't it? You sure can pick that up. And then there's another quote here. It says, smells kind of funny in there. What did you guys do? And there's laughter. And so this is after the EVA and the return back to the module, and they're smelling potentially a reactive sulfur component, and they're making jokes related to someone potentially passing gas. Now, in this part of the talk, I'm going to talk about or discuss data from two soils that we analyzed, 74241, that was collected on the edge of Shorty Crater, and 75081, that was collected on the edge of Camelot Crater. And just to give you a bit of perspective or scale on the distance between these two collection sites, they're about 3 kilometers from one another. Now, in my work and in original studies from the 70s, there was this interesting observation where there was a relationship between sulfur concentration, the delta 34S composition, which is that mass-dependent fractionation tracer, and also grain size. And the way I'm illustrating grain size in this figure on the left is by using an outer layer function. And what you can, in order to kind of put that in the reference frame of the size of grains, the largest grains would be at an outer layer function of 0, and the smallest grains are at an outer layer function of 1. Now, the original hypothesis for this relationship was that it was dominated or controlled by a fractionated condensed layer of vapor that's added to soil grains after gardening events. However, later it was also suggested that there is significant mass-dependent fractionation that occurs during diffusive loss of volatiles. But if we look at, say, if we try to model that original hypothesis, we find that that model can fit the isotope data and the relative to grain size, which is shown on the bottom here. But as recognized in the 70s, this is not necessarily a model that can fit the relationship seen between sulfur concentration and grain size. And so one of the most puzzling observations back when we started looking closely at this data is that there is a dip in the sulfur concentration as you go from large to small grains, and then it upticks again. And so this was one of the weird observations, particularly with the sulfur concentration, that we decided to look further into by performing some diffusive models. So we explore the role of diffusion, where we have sulfur being lost from the interior of a grain out to the edge of the grain and adding a condensed layer of vapor. And in our analytical models, we find that we can reproduce the relationships seen between sulfur concentration, isotopic composition, and grain size, but we also find that in order to fit the data, it requires two sets of parameters in terms of the time at which this process occurs and the starting sulfur concentration, which can indicate that there are different histories associated with these soils. Maybe it's related to their maturity. And another major observation that we found with our models is that the relationships that we're observing is not necessarily controlled by the process of diffusion, but instead it's controlled by however we've defined that condensed layer of vapor. Now we can simplify our models by just by using a 3-component mixing model where you have, say, an unaltered soil grain, you have sulfur loss towards the edge of that grain, and then you add a condensed layer of vapor. And what we find, again, is that that simpler model can reproduce the relationship seen. But what's different is that we now have constrained that it requires a thick depleted melt layer towards the edge of the grain, say about 20 microns, that loses 0.5 to 0.75 of the original sulfur. And even more interesting is that this process of sulfur loss from that depleted melt layer has to occur without any isotopic fractionation. So ultimately, from the modeling, in terms of trying to understand the relationship between sulfur concentration, the grain size, and isotopic composition, what we find is that we can reproduce the relationship seen, but it's not perfect. And we also find that potentially there's these two different soils underwent different histories in terms of their processing. And what's also an interesting finding is that this process of sulfur loss has to be occurring at the edge of the grains, and it's melt depletion, say at the edge of the grain, where most of that loss is occurring from the original soil grain. And lastly, our isotopic composition appears to be controlled by the condensed vapor component. However, our model still does not explain how that condensed vapor component was ultimately fractionated. All that we know is that it's a fractionated condensed layer vapor component. So to place constraints on how that condensed layer of vapor could have been fractionated, we could potentially look to the minor isotopes of sulfur. And this is a way to potentially first determine whether there's evidence of photochemical reactions that occurred, say if when the vapor was produced from the micrometeorite impacts. And if there was a photochemical reaction that we can observe in the minor isotopes, it could indicate that there's some complex atmospheric chemistry that includes kinetic mass-dependent fractionation through atmospheric escape. And ultimately, you could have mass-independent fractionation imprinted on the mass-dependent fractionation of sulfur loss of that vapor that's ultimately added to the soil grains. So if we look at the cap delta 33s, that mass-independent fractionation tracer for sulfur, versus the delta 34s isotopic composition. As I showed earlier, the delta 34s compositions are fractionated towards more positive values, say for 74241. But what we don't see is any evidence of a photochemical reaction that must have occurred for the condensed layer of vapor. And so what this tells us is that, I guess during this process of sulfur loss, even though it is a kinetic mass-dependent fractionation, it's not necessarily a process that's going to shift the cap delta 33s. And so that would be a much larger implication for other systems, such as volcanic degassing on Earth, where we're measuring small cap delta 33s variations, looking for crustal recycling, and also seeing processes of sulfur degassing in that system. And so if we use the moon as a natural experiment of kinetic sulfur loss, say to space or, in a vacuum, and we don't see any cap delta 33s variability, we can then say for other systems that if you have any significant degassing of sulfur, you should not see any changes in your mass fractionation law that could be misinterpreted as a, say, mantle recycled component or an exogenous component. When we look at the data from 75081, we see that it has a different cap delta 33s composition, and it preserves large mass independent fractionation. And what's also important to note is that that the data between cap delta 33s and delta 34s is negatively correlated. And I'd like to point out that in this plot, the largest grains are plotted as these squares with the cross through them, and the smallest grains are the squares without the cross. And so what this relationship is telling us is that we do have, we likely have mixing between two components, and that the largest grains preserve the original component or the original isotopic composition. It looks like it's trending towards the isotopic composition of lunar basalts. And the smallest grains are reflecting the isotopic composition of, say, whatever the condensed layer vapor component is that's added. Now, we could consider whether the cap delta 33s anomaly is from, say, the micrometeorite impactors, but it's hard to reconcile that given that we don't have a clear match with any other early solar system materials that we've analyzed for sulfur isotopes. So it's not necessarily easy to link these isotopic compositions to bulk solar system materials that could be bombarding the lunar surface. It's also, we could also consider that this mass independent signal was captured, say, from an ancient global atmosphere through basaltic and fire fountain eruptions. But although the constituents of the soils are old, such as the basaltic fragments, the gardening process is young. And additionally, if this was an ancient global atmosphere, we would expect that mass independent fractionation signals would be ubiquitous across the lunar surface. And as we see, especially for lunar basalts, it's not observed. Another potential way to produce cap delta 33s could be through, say, sputtering from solar wind interactions. But it's important to note that both of the soils we analyze have experienced sputtering from solar wind, yet only one of the samples shows mass independent fractionation. So it's difficult to then link, say, solar wind sputtering to the cap delta 33s composition. So ultimately, what we end up concluding on is that the nature of the mass independent fractionation seen in 75081 is linked to photochemistry, and that this photochemistry occurred during the production of a transient atmosphere during lunar gardening events. And we kind of anchor our hypothesis on the relationship between grain size and cap delta 33s, because it appears that as the delta 34s is increasing, and we know that that's an indication of mixing between a condensed vapor component on a small grain versus a large grain, that cap delta 33s is decreasing. And so they're likely associated with one another. And so our hypothesis is that as the grains are bombarded and sulfur is volatilized, these transient atmospheres, ultimately end up losing sulfur as a kinetic mass-dependent fractionation, which shifts your delta 34S value positive. And at the same time, you have sunlight bombarding that gas and producing mass-independent fractionation that is then captured and condensed on the soils. Additionally, I think it's important to note and discuss this difference between 74241 and 75081. Why does one soil show mass-independent fractionation and the other one doesn't? Now, what we know is that for, at least in the lunar environment, in order to produce cap delta 33S compositions, photochemistry is required. And photochemistry requires sunlight and gaseous sulfur compounds. So the difference between the two samples either reflects, say, a difference in sample maturity. We know that 74241 is an immature soil and 75081 is a submature soil. And so there could be, say, a difference in the amount of solar wind and planted hydrogen, which then could play a role in how much, say, H2S is ultimately released into that transient atmosphere and bombarded by UV to produce mass-independent fractionation. The difference could also indicate that there was potentially processing of these soils in distinct lunar environments, such as a sunlit versus shadowed region. So in our next steps, thanks to Solar System Workings and Ryan for approving our request, our plan is to analyze a suite of soils that are varying maturity. We're going to again sieve our soils and perform high precision sulfur isotope analyses, including a repeat of 15021. So if we look at the original measurements by Rees and Thode in 1979, we see that potentially there is some sort of relationship between the immature 74241, submature 75081, and as you go to the mature, the delta 34S is increasing, and it appears that maybe the cap delta 33S is more heterogeneous. That could indicate that maybe the soils are being processed over and over and the gas is going up, interacting with sunlight and condensing over and over again. That amplifies that cap delta 33S composition. But again, this is just a hypothesis, and hopefully with the help of my two incoming graduate students and my current research lab technician, we can place some constraints on what is causing the cap delta 33S compositions in various lunar soils. Now, when we think of where, like in the event that it is, sorry, I have pound signs there. I forgot to change the sample numbers. But when we think about environments where it's dark on the moon, where you could potentially have soils being processed and not producing mass-independent fractionation, you could consider that the moon has day and night cycles. So perhaps one soil was processed during a lunar day and the other was processed during a lunar night. We can also consider that there are permanently shadowed regions on the moon. And so maybe one soil was processed in a permanently shadowed region. Perhaps in, say, like in part of a crater as a micro cold trap. And what's important, one of the interesting observations of our data is that there is, say, we have that strongly negative cap delta 33S value associated with 75081. But in order to close mass balance among the isotopic compositions, it requires a strongly positive component to be on the moon somewhere. And so this is a component that may exist, say, in lunar polar regions or condensed in other cold traps. We're not necessarily sure. And so there could be some additional effect of having permanently shadowed regions and micro cold traps in the processing of sulfur um, among lunar soils on the surface. And so in our next steps, we are going to be analyzing two different samples that are related to one another, where one soil is illuminated and the other soil is shaded by a nearby boulder. And so we would be able to use this soil as a natural experiment to determine whether there is a notable isotopic difference between these two samples, assuming that they were processed during, say, the same time. So to summarize this part of the talk and my hypothesis for how lunar soils and the sulfur within them have been processed, we think that it first starts with a micrometeorite impact, and you have sulfur loss from the edge of soil grains, and also loss of any additional sulfur that escapes with hydrogen. And as that gas is released from the soils, you end up having UV photolysis that occurs in these transient atmospheres that produce cap delta 33S variations. You can also have the loss of 32S from that atmosphere that would result in the increase in the delta 34S values. And finally, as you end up with a lunar soil with a condensed layer of vapor, we expect that the mass-dependent fractionations are going to be amplified with maturity, which is an observation that was made back in the 70s. But we also are considering that mass-independent fractionation is going to be amplified, or mass-independent fractionation is going to be amplified with maturity. And that has yet to be determined as of now. So now I want to switch gears and discuss a related but different project where I analyzed the bulk sulfur isotope composition of lunar volcanic glass. So for this part of the study, I focused on orange and black glass beads. Again, these glass beads contain this amorphous layer of condensed silicate vapor, in addition to quenched melt that could reflect mantle compositions. And the point of this study is to use the sulfur isotope compositions of these glass beads in order to place constraints on the sulfur isotope composition of the lunar mantle and determine genetic links, potential genetic links to Earth. But then it also offers an opportunity to assess whether mass-independent fractionation or photochemistry occurs during the fire fountain eruptions and the production of volcanic clouds. And particularly, we can use the quadruple sulfur isotope measurements in order to test the various hypotheses associated with establishing these orange and black glass beads. Now, I want to point out that there was a study previously published by my colleague Alberto and Eric Howry in 2021, where they analyzed the delta 34S composition of olivine-hosted melt inclusions and volcanic glass beads from a variety of sample types, orange, yellow, and green glass. And the reason for analyzing the glasses and melt inclusions was to try to place constraints on, or by using a sample type that captures sulfur, say at the time of eruption, before any further modification of that glass or melt inclusion occurs at the surface. Now, what they find is that they see significant delta 34S variability from, say, +1 all the way down to -15 per mil. And they link this delta 34S variability to processes of sulfur degassing. And now what's unique about sulfur degassing models is that we use them in order to identify what our starting compositions could be. And what what Saul and Howery found in 2021 is that the various glass bead types, such as orange, yellow, and green, appear to have different starting compositions, which may be linked to, say, different reservoirs of sulfur in the lunar mantle established by mantle or core formation and differentiation, also melt production. But what this study does not have is the cap delta 33S values associated with the different soil types. And although the delta 34S starting compositions may be an indication that there are multiple sulfur reservoirs in the lunar mantle, it's not clear whether these reservoirs were established by typical mantle differentiation processes or if it's linked to a heterogeneous distribution of sulfur that was delivered originally to the moon. So as I mentioned before, I wanted to use sulfur isotope data from lunar glasses in order to test for genetic links between the Earth and the moon. When we look at the cap delta 33S composition of lunar basalts relative to estimates for the bulk silicate earth that's been derived from mid-ocean ridge basalts, what we find is that at least between Earth and the moon, the cap delta 33S averages are similar, which could indicate that there was efficient mixing, say with the giant impactor, or the impactor had a similar composition as the proto-Earth. But what's interesting here is that the delta 34S compositions are different between lunar basalts and bulk silicate Earth. And now it's important to kind of make clear that although the delta 34S values or compositions are different, that's not an indication that there is not a genetic link between the two. For a genetic link, we specifically need to assess the cap delta 33S composition. And this is because the delta 34S compositions of the Earth and the moon have been altered. So for Earth, it's been shifted towards negative values, either through core formation or accreting fractionated precursors. And for the moon, we're not necessarily sure why it's shifted more positive, at least for lunar basalts. Perhaps it's from melt generation or degassing of sulfur during giant impact or lunar magma ocean. But I just wanted to point out that there is this difference, at least in delta 34S. So as I mentioned before, I analyzed both orange and black glass beads, and this is from a drive tube 74002/1. And what we observe for, again, sieved glass beads is that we have a spread in delta 34S. But instead of going towards more positive values as grain size decreases, we go towards more negative values. So the largest grains are here, and the smallest grain size, which is less than 10 microns, is this round data point here. And I'm realizing that I mixed up my symbols, but it is correct in the published manuscript. So what this indicates is that we do have two different components associated with the orange glass beads. One that is more fractionated in delta 34S, associated with the smallest grain sizes, than the delta 34s composition of the largest grain sizes. And the largest grain sizes appear to share a delta 34s composition that is similar to lunar basalts, which would suggest maybe, however, that, I guess, orange glass melt source was established or originally established through differentiation was potentially associated with the same process in establishing the delta 34s composition of the lunar basalts. However, when we look at the cap delta 33s composition of the orange glasses, what we find is that we see no difference between the isotopic composition of the largest, the cap delta 3s composition of the largest grains and the cap delta 33s composition of the smallest grains. And so, considering that we have a relatively consistent cap delta 33s composition across all grain sizes that is distinct from the origin, which we established as 00 from Canyon Diablo troilite, and it's distinct from the bulk silicate moon estimate from lunar basalts, this is an indication that we don't necessarily have evidence of mixing with a photochemical component that's similar to the same way we see mixing with the lunar soils, where we saw, a negative correlation between the delta 34s values, cap delta 33s, and grain size. So considering we don't have any clear evidence of mixing with the photochemical component, we then have to ask ourselves, well, what's the source of this anomalous signature, right? So in order to we first have to step back and take a look at the, say, degassing models and whether they provide any evidence that we are actually getting a condensed vapor component that is mixing with the various grain sizes. So when we perform a degassing model similar to fractionations that were published in Saul and Howery 2021, we find that the composition of the melt falls through, at least for the orange glasses, it falls through the majority of the largest sieved grain sizes. But the smaller sieved grain fractions don't plot on the composition of the melt. And we interpreted this lack of plotting along the same degassing trend as the addition of a condensed layer or vapor component dominating the isotopic composition. So although you are fractionating, that vapor component is following the dashed line. As it gets added to the smaller grains, or the surface to volume ratio changes as you go towards smaller grains, that isotopic composition is more pronounced. And that's why we don't see those small grains falling along or plotting along the melt degassing trend. I'd also like to take this opportunity to point out that when we analyzed the black glasses as a bulk, say, as the bulk sieved measurements, none of those samples fall along the melt degassing trend. However, when we performed our leaching experiment, we find that the interior, or the glass beads, have isotopic compositions that do fall along the composition of the or the model for the melt degassing, or the composition of the melt. And the vapor or condensed vapor component is much lighter in delta 34s, and we see that it does not, again, plot along the composition of the melt during this degassing process. And so what's very clear here is that we have two components. One component of sulfur that is trapped within the interior of, say, these glass beads and a vapor component that's condensed on the outside. So if we want to, say, figure out where is this cap delta 33S composition coming from associated with the orange glass beads, if it's not from a photochemical component, at least during the time of eruption, we can then guess or ask ourselves, could it be from entrainment from surrounding crust? But likely it's not, because the rates of entrainment during that eruption are likely too slow. Perhaps there was ingassing of mass-independent fractionation into the glass beads, and that's how we're able to decrease the cap delta 33S towards lower values relative to lunar basalts. And this is a hypothesis that could potentially be reconciled with the model for how the orange and black glass beads were established, where you have orange beads condensing on the outside of the cloud, and the black glass beads kind of cycling through this vent, becoming more devitrified, more degassed, and considering that there's this stratification within the cloud of the orange and the black glass beads, it's possible that mass-independent fractionation could have penetrated the edges of that cloud, and that the black glasses in the interior were just too deep in the cloud, where it was not optically thin enough for UV to penetrate, ultimately resulting in differences in the cap delta 33S between the black and the orange glasses. And so there was an LPSC abstract that was recently presented on, where there's an argument where there is actually ingassing of sulfur, which is a really unique finding for orange glass beads, where the idea is that there's possibly up to 20% ingassing of sulfur into the glass beads. However, what's not clear about this observation is whether it excludes the need for a mixing relationship in triple sulfur isotope space to explain our cap delta 33S data. If there is still 80% of that sulfur in the smallest grains versus the largest grains, what would produce the differences, say, in delta 34S, but maintain a cap delta 33S? And so that is, say, an ongoing question that should be looked into. So we've ultimately reconciled our cap delta 33S best as a source feature. And so in order to have well, I guess considering that the moon does not have plate tectonics like Earth to bring surface material into the lunar mantle and back up again via recycling, this would require some sort of different process to put material that was once, say, in the lunar atmosphere into the lunar interior and back out again. So perhaps this is mass-independent fractionation that was captured early in lunar evolution. Say perhaps it occurred during lunar magma ocean phase, and say that sulfur was deposited or rained out as elemental sulfur on proto-lunar crust that then delaminated or was brought down during mantle overturn. Perhaps this is some sort of primitive recycling on the moon. It could also be related to, say, the injection of some sort of exogenous meteoritic component. There are CM chondrites that have sulfur phases with anomalous cap delta 33S. But if you do have some sort of injection of an exogenous component, it would require that there's inefficient mixing with the rest of the mantle. And so we'd have to come up with a reasonable explanation for why we not have efficient mixing with these different components. It's also possible that it's a primitive signature. Maybe it's real, and that the Earth and the Moon started or were established with different cap delta 33s compositions. If we look at the cap delta 33s values of iron meteorites and main group pallasites, we see that different potential parent bodies have different cap delta 33s compositions. And so, although we have not found this, say, on Earth by sampling primordial reservoirs, it's still possible that there are solar system materials, including Earth, that has sulfur trapped within its interior that is mass independent or different from what we would assume is the origin. And at least in the lunar system, it's possible that we're only seeing this in the orange glasses because the rest of the mantle was later altered, say by late accreted materials that shifted the delta 34s composition closer, or the cap delta 33s composition much closer to zero. And that is possibly why we see that homogeneity in the lunar basalts and our heterogeneity with these lunar glasses. But ultimately, we don't know. We don't know where this cap delta 33s composition comes from associated with these orange glasses. And so it requires more work. And this is work that we plan to do related to CERVI, CASA Moon, where we're going to be analyzing many different isotope systems of different portions of the drive tube 74002/1 from the top all the way to the bottom to really try to understand the nature and history of this volcanic eruption and whether we can gain more insight into how volatiles were processed during this eruption. And particularly, especially when we look to the absolute at the topmost part of this core, which is a different sample, 74220, versus the bottom, if we see distinct differences in how these materials were processed, because this could provide additional insight, especially with those in-gassing models, with how much sulfur is in-gassed and the different environments related to how processed these materials are. And I'd also note that with my original study, I did not perform a leaching experiment with the orange glasses, and so that's going to be performed with this future work. And we also plan to collaborate with a sister survey, Lunascope, who's performing atom probe tomography of condensate on orange glass beads in order to determine the composition and various phases of sulfur on these bead surfaces, which would ultimately aid in how we set up our sequential extractions and which phases we hope to extract out. So with that, I will leave 2 images of my hypotheses for the different parts of the talk and take any questions for the next 6.5 minutes.
Thank you very much, James. Are there any questions online? If you do have a question, just spit it out. I'm not going to call on you.
Hey, James, this is Francis at JSC. Really nice talk.
Thank you.
So you had mentioned at the end that you're going to look at other isotopic systems, and I had a question sort of geared towards the first part of your talk about what other isotopic systems could help you narrow down the process of whether this is like a, shadowed soil versus sunlit soil or other type of process.
Yeah, so I think I said I'm gonna analyze. No, it's the whole lunar scope team I'm not gonna do any lead or any crazy clean lab chemistry like that. But I think that in the 70s they were analyzing, they looked at the relationship between nitrogen and sulfur for sputtering effects. And so that would be like a nice way to try to identify how sulfur is fractionated during solar wind interactions. Um, but then also with kind of trying to identify those exogenous components that could exist among the lunar soils and glasses. There are different, the different isotope systems that look at nucleosynthetic effects. I think like chromium would be one. And, those systems like that where it requires really high precision measurements of these rare isotopes that could help with our identification of exogenous nucleosynthetic components. And then my class, I think it was on, yeah, Monday, we talked about triple oxygen, and I don't know how much, I'm sure Tony and Zach could shed light on this, how much triple oxygen isotope measurements were performed on sieved glass beads, and if that could be any indication of primitive or ongoing mass independent fractionation if you're looking at CAP 17O across different grain sizes.
And we got a question from Zach Sharp.
Yeah, hey, James. Thanks for that nice talk. You have a plot which looks like a Rayleigh distillation. Maybe you could go back to that for us. It's about 5 backups. And there are samples that have a low delta 34S, but a higher F value, like 0.2 or something.
Yeah, let me try to get back. I'm not used to WebEx. Hold on. Yeah, here we go. Okay. And you're talking about this one.
That's right. So the one that's down there at a fraction of 0.2 and lies below the Rayleigh fractionation line, if those samples couldn't be, the gaseous phase, which I gather is heavier than the source, but still, as you get to a low fraction remaining, the gas itself will be light. And if that couldn't be adsorbed onto the surface, which will increase the apparent amount of sulfur and therefore give you a higher fraction remaining value of 0.2. So it's actually a mixing of a really light value with a more unaltered sample. So like a kind of a straight line between something in the lower left corner and in the upper right.
Right. Oh, you're just making a comment?
No, I'm just asking if maybe that's what those lines are, that you have those horizontal arrows.
Yeah.
Adding light material, which is apparently increasing the fraction remaining value. And it's not actually that, it's just addition.
Yeah, it's just addition of a specific composition. And I guess I oversimplified it because I am still looking at like the relative proportion of grain size to component. But I don't, maybe it could also be that like the grain sizes for the leaching experiment need to be adjusted because it's not grain size, but it's rather the thickness of that layer. And but like you said, straight down, I mean the black data points are the bulk ones, right? So if like it closes mass balance, like if I do like a mass balance calculation between the gray red points and these ones up here, they fall right in the middle. So there is some sort of straight up and down mixing. Like you said, it could be like a super light value, and then a more heavy value here, and then we come up in the middle. Yeah.
No, I think those horizontal dash line arrows, is that the mixing that you're talking about? That they go from the vapor curve to the right and then intersect your measured values.
No, I don't think that's the mixing. I think
That's what I'm asking.
That's just me illustrating.
If you have vapor that's, let's say, originating on the dash line at -2.5. And then just add that to something, let's just move it over to the right, because you're adding material, you have an apparent higher fractionation remaining. Yeah.
So yeah, I mean, I think I'm just probably maybe oversimplifying, or maybe I don't know. I'm just trying to illustrate that the vapor component is just dominating the smaller grains, and it's showing us that we have two components. So if I really wanted to mix, it would be something like that. And maybe what I should do is plot like a 1 over S concentration versus delta 34S and demonstrate linear correlation to show mixing. Yeah.
Okay, good, thanks.
Sorry. Yeah.
Well-
I'll try to think about a better answer.
James, well, thank you very much, and we've run out of time. And I just wanted to thank everybody for joining us today. And next week, we will have Carl Agee, the director of the Institute of Meteoritics, who will be speaking about some exciting planetary sample materials that have been that are in the UNM IOM Meteorite Museum and the science that has been done on them. But again, I want to thank everybody for joining us and see you next week. Bye-bye.